Good afternoon, everybody, and sorry for the technical problems. My name is Yanli. I'm from Penn State College of Medicine at Hershey, which is the sweetest place on earth. And I'm from Dr. Feng Yue's lab. And today, I would like to thank the organizers for giving me the opportunity to present one of my projects, which is visualizing three-dimensional organization and long-range interactions of the mammalian genome with the 3D Genome Browser. So as you guys already have been familiarized with within this session, HiC is a chromatin proximity ligation method that captures unbiased genome-wide pairwise interactions within the genome. And it begins with cross-linking of a pair of loci that are interacting, ending with a biotin pull-down of the pair that hopefully could be sequenced in para-end sequencing. So HiC data are represented as contact matrices, as you guys all know. And given this pair-ended HiC data, if we map each pair-end into the genomic location, we can bin the genomic location into individual loci, such as the size of each loci is equal to the resolution. And this is different from the previous talk, in which each bin is actually a restriction enzyme, but here it's just a constant resolution. And as you can see, the contact matrix is reflected diagonally such that the contact interaction of loci ij is equal to loci ji, which makes sense biologically. So, but unfortunately, HiC data, as well valuable as they are challenging for the average biologist to analyze and interpret them. And one of the reasons is that unlike the other epigenomic data that are studied, that are stored in bed files and GTF files that are big O of n, due to the pairwise interactions as well as the matrix structure of the HiC data, it has an intrinsic complexity of big O of n squared. And these are just some hard numbers to really make this problem concrete. So with the advancement of HiC, when we increase the resolution of the matrix by 10 to the 3-fold, we increase the number of data points by 10 to the 6. So as you can see, this poses a lot of challenges in storing, transferring, and analyzing the data. And the other problem in data interpretation of HiC is challenges in data integration and contextualization. And of course, you guys are aware that this is a problem that's actually a problem in the entire field of bioinformatics, not just HiC. So there are actually some current HiC browsers that have tried to address these issues in their own way. Most notably, the Juicebox, which is released as part of the Raw et al. Paper by Lieberman Aiden's lab, as well as the WashU Epigenome Browser from Dr. Ting Wang's lab. And unfortunately, as beautiful and as these HiC data browsers are, and they provide their own contextualization, they really fall trapped into the big O n squared problem. So that's why we propose our own browser, the 3D Genome Browser at Penn State, which has these features that are to address these two problems of both to address problems of HiC data and to really increase the accessibility of HiC data to I guess, the general scientific community. And I'll highlight some of the features as I go on. So to start with, our 3D Genome Browser hosts updated HiC data sets. And we have the most comprehensive sets of data compared to the alternatives, especially the Raw et al. One, which have the coveted 1 KB resolution GM cell line HiC data set. And we're also excited to add Drosophila species datasets from the Victor Corses lab, and as well as potential zebrafish datasets. So it's no longer a mammalian genome browser anymore, but we're really excited for the next milestone. So the second feature is that the 3D genome browser allows searchable genomic features. And so what this means is instead of having to provide a location of the genomic feature of interest, you can actually distill that to enter a symbol that is actually biological meaningful, such as a gene name. And this seems like a trivial feature to point out, but you'll be surprised that the current high-C browsers do not have this feature available. They're all location dependent. So, and one of the highlights for our browser is that this search field is actually linked to the MySQL tables from the UCSC, University of California at Santa Cruz Genome Browser. And if you are familiar with the MySQL database from the browser, then you know that it's very comprehensive and flexible to use, and this is what we're trying to take advantage of. So for this example, we entered the gene for Sawnage hedgehog. Okay, so we entered our gene, and here's what we get. We see the rotated high-C map, which is, as you know, that since it's reflected diagonally, we only offer the one half of it to really save some space and computation power. And we have contextualization from the UCSC Genome Browser. And for those of us who use it, you can actually see the familiar interface here. And then the good thing about this is that you can interact with this UCSC Genome Browser as you would normally with a website. So you're free to modify tracks, add tracks, and use your own UCSC session. So we have our Sawnage hedgehog gene here, and this is when we see a couple of tags surrounding the gene. And we would like to explore more of the data so we can navigate either with these buttons up here, which could traverse the data or zoom the data based on your preferences, or you can actually just directly double-click on the region of interest. So once we do that, we can see that we have a blown up of a region. Looks like a tad. And then we could manipulate the intensity values to really highlight high-C peaks, the signals that are not noises. So here we can adjust our intensity cutoff value, and we can see that this is indeed a high-C peak. Okay, so what does that peak mean? So contextualize, contextualize, contextualize. So we have our UCSC browser, and I have added tracks that are pertinent to this example here. So here is the, and this is really difficult to read. So you could single click on this on your region of interest, and you can see there's a shader that comes out that highlights the region. So you can see that here is a Sawnage hedgehog gene, and this is the exact point where the high-C peak is. You see the Sawnage hedgehog TSS right there, and then you see an intron, the intron 5 of LMBR1 gene. And it's hard to see here, but there's actually a yellow peak for H3K4ME1. So it indicates it's some sort of cis-regulatory element or something that is actually physically interacting with the Sawnage hedgehog gene. And I would like to stress again how great it is to interface really with UCSC, because a lot of online resources interface with UCSC and provide their own tracks. So here I enabled the Decipher track. And then how to read this, if you're not familiar, is that each bar here represents a single patient. And the red is deletion, and while the blue is duplication. And as you can see, surrounding this region, there is a blue bar here. And this patient that has a duplication in this region has some sort of polydactyly phenotype. And excitingly, this is actually biologically relevant and consistent with literature. So actually, it is true that intron 5 of the LMBR1 gene has an enhancer that affects the Sun-Edge hedgehog gene. And we know this because the disruption of this gene, of this enhancer, actually causes the malformation of the limbs in mouse through knockout. But if you have some sort of duplication of the enhancer, you actually have the preaxial polydactyly phenotype, which is an autosomal dominant disease. And what it means is basically you have an extra finger toward the thumb or toward your tibia. Okay, so that's all well and good. So what we consider to be really the highlight of our browser is that our 3D Genome Browser supports browsing of external and user-generated datasets. And how we accomplish this is that we have pioneered a new format to store contact matrices called the BUTLR format. And the BUTLR stands for Binary Upper Triangular Matrix. And trust me, I've gotten a lot of flack for this name, but it's the best one I could come up with. So how this file format works is, of course, from the name you can tell it's a binary file. And then it stores the pointers to each row in the header. And each row, each record in the row actually stores a pointer to each column. And so this really supports random access of the file. And then since the offsets for these records are constant, we can do some pointer arithmetic to go directly into the row of interest and then retrieve the column data. And so this really cuts the big O of n squared into big O of n. And then we actually also aim to minimize redundant data. And the way we do that is only the upper triangular data is stored. So where I of ij, where I is less or equal to j, that part is stored. And then we also remove the most common values, and that's usually 0 for most high-C matrices. So these are not stored. So this really compresses the data. And here is a graph of the contact matrix resolution versus how much memory we need to store the data. And as you can see, this is around 50 KB. The data for just normal tab delimited text file just blooms up according to the big O of n squared complexity, while BUTLR files scales much better. And then less than 50 KB, the two file sizes are comparable because of the storage of the row and header for the BUTLR files. But the most exciting part is not actually the compression part, but the random access. This allows us to store the BUTLR file in an HTTP accessible server. And we can, on our browser, could directly query the relevant regions to display in the browser instead of having to upload the entire data set. And this is similar in concept to the big WIG and big bed files of UCSC Genome Browser, if you guys are familiar with. And then here is just an example. So this is the assembly of NA12878 that is really constructed by this group here. And so just by entering the BUTLR data, as well as the URL for a UCSC Genome Hub that are created directly for this assembly, we could visualize the high-C data from new assemblies. And this is important, as you know, UCSC have moved on to HD38. So if we don't have the chance to remap the data, then people could do their own mapping themselves. Okay, so, and then the last feature is that the Genome Browser optimizes the interpretation of local interactions. And the way we do that is actually we have a virtual 4C plot. And a 4C plot is a plot, just a plot of the number of interactions between a single locus of interest and its surrounding loci. And it's virtual because it's derived from 4C. It's not actually, derived from high C. It's not actually the 4C experiment itself. So as an example, we have a SNP that we know is implicated in increased levels of LDL, as well as myocardial infarction. And we could enter this SNP of interest into the virtual 4C browser. And we can see, here's our virtual 4C browser. The bait loci, which is our SNP. And then we can also click on things that we find interesting. So we see that here, this is some sort of cis-regulatory element, as you can see the histone peaks. And then here is actually the TSS for the SORT1 gene that actually goes this way. So here's the TSS. And then if we want to navigate this more, we can double click on this region to center it. Wait, sorry. To center it here. And then here is the SNP is actually here now. And we can see that the peak holds over here as well, even if we change the center. And we can see that with UCSC Genome Browser shows that this overlaps with a CEBP beta transcription factor which binds here, and this is available in liver. So this is also have G2 data. So this is consistent with the biology that that this SNP affects the binding with CEBP, and it modifies the expression of the SORT1 gene, and that's causes the disease phenotype. Okay, so hopefully by now I have convinced you that our 3D Genome Browser increases accessibility of high-C data, particularly for local interactions, and that it's going to be a valuable tool to drive hypothesis generation, as well as guiding biological validation experiments, particularly for when studying the GWAS implicated, the functions of GWAS implicated non-coding genetic variants. And I would like to thank everybody in my lab, as well as other labs who made their data available. This project is always under construction, so we will welcome any feedback. Thank you very much.
So thanks, and this is very useful. I can attest that it works. I've used it myself. And I had a couple, maybe they're just feedback.
Yeah.
I don't know why you're showing somebody else's slides.
Yeah, I'm not sure. I'm not sure why either. What is?
But one question is, you said contextualize, context 3 times in a row, really fast. I don't know how you said that. But it strikes me that your display, you do this this triangle, right, in your high-C context, which has half of your display is white, and you're losing. You have all that space that you could use. I know that the triangle shows the context that involve the locus that's shown below. But it strikes me that the neighborhood contexts might provide useful information. If you saw a big trend over there, you might move your browser to the right to find out what's going on. So I think there should at least be a toggle where you could turn on those guys and maybe leave some kind of line that shows where the triangle is. But it seems like that those flanking regions could be informative to the user.
Okay, definitely.
And then I had another suggestion, but I forgot what it is. So anyway, I don't have a question.
You can talk to
But thank you. It's a good resource.
Picking up on the same track, maybe this is your last suggestion. Do you have a toggle to overlay previously annotated TADs so that you don't have to kind of like maybe squint at a triangle and hope that suggest that it's a previously annotated TAD, or maybe it is, maybe it isn't.
Or confidence estimate.
Or a confidence estimate. Oh, see, community.
What is it?
I'm wondering if like you could have a toggle to overlay previously reported annotations of TADs.
Oh, okay. Yeah, we could work on that. But right now, you can actually upload TADs as a bed file.
Okay.
In the meanwhile, but yeah, we can definitely think about adding those.
Well, right. Lots of them. Cool. Thank you.
Mm-hmm.
Any other questions or suggestions? Okay. Thanks, speaker.